Now, after studying about the biotechnological applications in agriculture, let us study about its application in another field, that is medicine. Most developments in biotechnology that is originated for potential application in healthcare. That is, biotechnology plays a very important role in the development of healthcare, and thereby it helps in the production of various medicines through biotechnological techniques. As a result of this, people can protect themselves from dangerous disease. The field of biotechnology has given various techniques like gene therapy, then genetically engineered insulin, apart from that, recombinant DNA technology, we have already started in Unit 11, that is its principles, tools and processes. Apart from that, PCR, that is polymerase chain reaction. Which helps in the production of oligonucleotide primers. Thus, these are few techniques out of all which helps in the production of new medicines and helps in diagnosing diseases. Let us study each of them in brief. So let us begin with the first technique, that is production of genetically engineered insulin. As we all know that insulin is a hormone that helps in the regulation of sugar metabolism within the body. And therefore, it plays an immense important role in the diabetes. That is, improper ratio of insulin within the body can result in diabetes. As we all know that insulin natural production is produced from the beta cells of the islet of Langerhans of pancreas. Thus, insulin is naturally produced from these beta cells of the pancreas. Naturally, human insulin contains 51 amino acids. That are arranged in form of two polypeptide chains. In form of chain A and chain B. That is, we are depicting these two chains as chain A and chain B, within which the chain A contains 21 amino acids and rest chain B contains 30 amino acids. That is, the distribution of amino acids takes place between these two chains in such a manner. Now, the insulin that was used for curing diabetes was initially extracted from the pancreas of slaughtered cattle and pigs. Since they were extracted from these animals, it was definitely possible that it can cause allergic reaction to the humans, which was definitely a major disadvantage of this extraction from pancreas. And therefore, in order to avoid this disadvantage, in 1980, with the help of recombinant DNA technology, human insulin was first obtained or produced from the bacteria E. coli. And such a human insulin that was produced in bacteria is called as humalin, which was first developed with the help of recombinant DNA technology. In this manner, the insulin was synthesized as proinsulin. Thus, from this, the insulin synthesis begins from the proinsulin, which has an extra polypeptide chain which occurs in a stretched manner. So, it can be defined as a extra stretch chain, that is polynucleotide chain, which was termed as C, chain C. Thus, chain C was produced within the E. coli bacteria in the humalin, which helped in curing the diabetes. However, the C peptide or the C chain was not found in mature insulin. And therefore, artificially it was removed during the maturation of insulin. Thereby, in 1983, a scientist named as Eli Lilly that belonged to the company of United States produced two DNA sequences that were corresponding to chain A and B. of the human insulin that was introduced into the plasmids of E. coli. To produce insulin chains. This we have already studied earlier, that how does the recombinant DNA technology takes place. That is, with the help of insertion of the gene into the plasmid of another organism, it helps in creating such genes in larger amount with the help of selectable suitable markers. And therefore, such a gene was insulated or artificially inserted into the plasmids of E. coli to produce new insulin genes. Further, these separated chains, that is these two chains, were then separated and extracted from the bacteria, of course, and then combined by creating disulfide bonds within the human insulin. That is, in order to produce human insulin, disulfide bonds were added between such chains. This figure depicts the production of human insulin by the genetically modified technique. As it can be seen from the figure that an insulin gene contains two chains, that is chain A and B, which possess specific sites for replication, which is further inserted into the E. coli bacteria where it is cultured and thereby produced in larger amount, which is then separated and extracted from the cells, which together by combining through the disulfide bonds form a together stretched chain of C-peptide, which results into the production of human insulin.